welcome back. It's my great pleasure to introduce the next speaker and alumnus of our state university, Dr. Randall Nelson. Dr. Nelson is a research geneticist with USDA ARS. He's also a professor of plant breeding and genetics in the Department of Crop Sciences at the University of Illinois. Dr. Nelson received his bachelor's degree in agricultural science from South Dakota State University, master's degree in crops production and physiology from Iowa State University, and a PhD degree in plant breeding and genetics from University of Illinois. Dr. Nelson's research is focused on soybean germplasm evaluation and utilization. He is a curator of the USDA soybean germplasm collection, which includes nearly 22,000 accessions. This collection is the second largest collection in the world. As a soybean breeder, Dr. Nelson has released more than 20 high-yielding germplasm lines derived from exotic germplasm. His presentation today is Germplasm, the past and the future of soybean breeding. Please join me in welcoming Dr. Randall Nelson.
Thank you very much. It's a real privilege for me to be here and to share this symposium with you, and I thank you for that opportunity. I guess it shouldn't come of any surprise to you when I would agree that you have made an excellent choice for your symposium topic. I think relating germplasm to plant breeding is a very important relationship. As a curator of a relatively large germplasm collection, and as a plant breeder that's used that collection in more than 3 decades of breeding, I get exposed to a lot of perceptions about germplasm. And I guess my conclusion is that I think plant breeding in general and most plant breeding programs in particular would benefit from a greater knowledge about the germplasm that's being used in those programs, and probably even more importantly, the germplasm that is not being used in those programs. To honor the official title of this symposium, I'm going to spend a little time on the history of how we got to where we are in terms of soybean germplasm and what I like to call is the commercially used gene pool in the U.S. But I'm going to spend most of my talk talking about the importance of expanding that genetic base. I think for soybean breeding, that's a critically important issue. And so I want to use this occasion to be on my soapbox, if you will, about that. I think the data now is overwhelmingly in support of the inclusion of that germplasm, and I think it's absolutely essential if we're going to make the kinds of progress that we can make and to meet expectations for the future. So, almost 20 years ago now, out of Tommy Carter's lab in North Carolina, they put together a very detailed explanation of the genetic base of U.S. soybean production. And he did this by going back and looking through the records and cleaning up all of the pedigrees of 80 years of soybean breeding. And so if you're have any familiarity with soybeans, you probably have seen these numbers before. So there are about 91 ancestral lines that have made any contribution at all to U.S. soybean production, and many of these would be a single gene for disease resistance. Only about 1/3 of those make any major contribution, and then a much smaller subset of that provide the majority of genes in our U.S. varieties. This pool was mostly completed by 1950, and there hasn't been very much added in the last 45 years. Now, since they did this work 20 years ago, I think we've learned a little bit more about our gene pool and maybe some of the practical aspects of plant breeding with that. So for me, I'm mostly concerned about the northern U.S., and that's a little different than the south. And what we know then is that some of the original lines that were included in that first paper ended up contributing to genetic dead ends. So they were used as a parent once, but then those progeny were never advanced on, and so they really didn't contribute to what we have today. With some more detailed genotypic characterization, we know now that a couple of those major ancestors were actually genotypically identical. So they didn't make a different contribution. And then, so if we look at that, well, and then some of them contributed only to the north, some only to the far north into Canada, some just to the south. So if we look at what's available to soybean breeders here in the northern U.S., there are only 25 ancestral lines that account for 96% of the genes, based again just on coefficient of parentage. 10 of those lines provide 83% of the genes, and 63% from 4 ancestors. So we have a very narrow genetic base. And I think the other thing, if you go back and look at the history, you'll find that almost without exception, all of these ancestral lines made their contribution through one cross. So not only we have a very few major ancestors, but each of these ancestors only had the ability to contribute half of their genes. So how did we get to here? For soybeans, we have what we call the uniform tests, which is probably somewhat similar to what Stephen was talking about with his wheat varieties. So starting in 1939 in the north and 1943 in the south, USDA organized the soybean breeders, public soybean breeders, at that point, they were only public soybean breeders, to cooperatively test their best lines. So if we go back and look at, say, the first 15 years of those data, we can see what was originally tested and how we came with the kind of ancestral lines that we have. During those 15 years, there were about 900 lines that were tested. Most of those were the products of intentional crosses, but about 150 of those were direct introductions from Asia. Some of these were the first cultivars that were grown in the U.S. About half of those were actually used as crosses, and we see their progeny showing up then in tests and in future tests. And out of those 86 then, not quite half of them actually became part of our genetic base that we have today. And 17 of those 37 were part of this major 35 that contributed 95% to the total U.S. gene pool. The remainder of those that contributed very small amounts were largely selected because they had genes for mostly disease resistance. Get my watch out here so I can see how I'm doing on time. So if we go to the next decade, in the 1960s, there were 13 soybean breeders in all of the United States. By the end of the 60s, plot combines were the new technology. And for a variety of reasons, program capacity was very small in those early years. So if we look today, we have almost twice that many soybean breeders in the public sector, not counting all of those in the private sector. We probably have more bioinformaticists and molecular geneticists than we have breeders. There are enormous advances in field equipment that allow us to plant and harvest at unbelievable rates. Incredible capacity for data management, DNA and RNA technology have all made enormous improvements in the capacity of soybean breeding in the U.S. I don't have the specific figures, but I suspect that it's not out of reason to say that one reasonable sized program in commercial industry today can probably accomplish more than all of the soybean breeders could in 1970. But what hasn't changed during this time? Besides our genetic base, our annual rate of gain. This was a study that was led by my colleague Brian Deers at the University of Illinois, and we picked out varieties that were released in the decades, beginning in the 1920s, going through the 2000s, Obviously, the first ones were all either introductions or developed by public breeders, and then when we get in the later years, we also included some private varieties in that. And I'm going to show you then the data from maturity group 2, 3, and 4, and this is maturity group 2. And the best fit for the data in all of these was not a straight line, but you can see the dotted line there is when there seems to be a flex. And so what we're finding in this case from 1968 on, there was an increase in the rate of gain that was slightly better than what we had previous to that time. Maturity group 3, that break point is in 1964. Maturity group 4, it's in 1971. Now, the confidence interval around that break point is not very precise. So it's probably accurate to say that probably sometime in the 1960s, about the time that scientific plant breeding really got going, we started to making more gains than we were before then. But with all of those advances that we've made in the last 20 years, our rate of gain has not changed. So a few caveats. Obviously, we need more defensive traits now than we needed in the 1960s. There have been some changes in the gene pool. Mostly this has come from releases in the public sector, which are bringing in new germplasm. But because most of those programs are sort of either at the fringes of the major soybean breeding areas, or those varieties have limited planted to acreage, in terms of affecting the major soybean production, those changes are very small. But the one thing that has changed is that we now have the resources to make major changes in terms of our germplasm. And what I'm going to do now is, for most of the rest of the presentation, is talk about those changes. And I'm going to highlight a lot of the work that we've done in our program, but also in other programs, to show that by using exotic material in our breeding programs, we can change things as fundamental as yield. And I think it's been a tough sell. Germplasm has always been used for qualitative traits, particularly disease and insect resistance. But to say that we can bring in new material and increase the yield of our elite lines when all of these land races or varieties from other places are much lower yielding than what we currently have has been a difficult sell. So to talk about this, I'm going to use the gene pool concept. We heard a little bit about Harlan and DeWitt in classifying sorghum. They also developed this gene pool concept back in the 1970s. And this is an indication of how easily species will cross with one another. So for soybeans, we have two species in the primary gene pool, the soybean and the wild soybean. And I'll first talking about using exotic soybean germplasm. And when I use exotic, the opposite of exotic for me is domestic. So the exotic doesn't necessarily mean strange, but it just means it's not domestic. So exotic could be a wild relative from Australia, or it could be a cultivar that was released from an experiment station in China 15 years ago. But both of those are likely, if not proven, to have genes that are not in our current gene pool. So for talking about exotic soybeans, I'm going to use data that comes from mostly last year's uniform test. We have a prelim, which is the PT and the uniform test. And so this is data over 24 environments in 2013 and 14. Iowa 3048 is the Czech variety, the best variety in there. This experimental line from our program was 3 days later in maturity, but you can see almost 3 bushels higher yielding than the best variety in the test. It has a pedigree that's 52% exotic. And again, when I'm talking about these, that 52% is based not on actual genes or markers, but is based on the pedigree. And that 52% is contributed by 8, and I'm breathing here, PIs, or plant introductions, or exotic material out of the collection. The uniform test 4, again 17 environments. Here's another line. It actually is from the same pedigree. It was 3 days earlier than the best variety in the check, and you can see nearly 4 bushels an acre higher yielding. Both the uniform 3 and 4 test, these experimental lines were the highest yielding entry in the test. Not only better than the best check, but better than any other experimental lines that were included. The preliminary 4 test, and this is where the varieties first come in, or experimental lines come in, was really an anomaly, but perhaps is a signal of the future. 31 of the 34 entries in this test were derived from exotic germplasm, and 24 of those 31 entries exceeded the yield of all of the check varieties in the test. This was the highest yielding check variety. This was the best line, almost 7 bushels an acre higher yielding, 2 days earlier. 50% of its pedigree is from exotic material. These 2 lines, about 6.5 bushels better, 25% of their pedigrees, and 2 to 6 days earlier. And then 2 more lines, 5.5 to 6 bushels, 5 to 5.5 bushels, about the same maturity, about 30% of their pedigrees. I picked these top 5 lines because all of these are 10% or greater higher yielding than the best check variety in the test. The other thing is that all 5 of those lines have a different pedigree. So those top 2, again, the first one had 2 exotic parents in its ancestry, the second one 4. These were 2 lines that were developed by material that I had supplied to Bill Shapaw in Kansas. And you can see they have 4 and 5 exotics. And this is material that Grover Shannon developed from material I supplied that has 9 PIs. If we look at the unique exotic parents in these 5 lines, there are 16. All of these are 10% better than the best check in that test. And when you think about northern soybean breeding has 25 ancestors, we have 16 in here. I think you can see that there's enormous possibilities here. The other thing that I like to point out is that most of these are maybe 1 to 2 generations removed from their exotic parents, whereas that would put us back in regular conventional breeding, somewhere back in the 60s and the 70s. So not only do we have new germplasm with high yield, but we have an enormous possibilities here for recombinations that have not yet taken place. If we looked at all of the uniform test data from this last year, and we looked at those experimental lines that were in there that were at least equal or better than the best check, we'd get 28 exotic progenitors, or nearly enough to double the potential gene pool from China, Japan, Russia, France, Morocco, Romania, and Yugoslavia. Now, I think oftentimes we think soybean germplasm from France and Morocco, you got to be kidding me. These particular from Eastern Europe was importing soybean germplasm from Asia, early part of last century, the same as we were. And what we found is some genotypic analysis of some of this material, even coming from Morocco, is genetically very distinct. Not because it originated in Africa, but because it was brought there from Asia and could be very different than what we got, even though they came about the same time. So as we bring in this alternative material, and we want to compare it to our conventional gene pool, what we would like very much to be able to do is to say, this is unique. We can declare it unique. And I would have thought at this point in my career we could have done that, and we still can't. That's a frustration. So I think one of the things that we learn is that breeding always outpaces genetics. We can do things that we can't explain. That may be a statement, but it's not all that satisfying. So one of the things we can do is we can try to map QTL that are coming out of this exotic material. And we've done that repeatedly. That shows us that whatever is increasing yield in that particular exotic accession is different than whatever we've crossed, whatever commercial variety we've crossed that to. But what it doesn't tell us is that there's somewhere in the commercially used gene pool that those genes exist, and we just haven't found that. The other thing that we've been able to do is to confirm a yield QTL. And so it's one thing to map them the first time, it's a second to be able to show that they do function in a different genetic background. And I show here then all of these environments in which we tested for two reasons. I think what's really important is that in all of the individual environments or combining over years or locations or all possible, this yield QTL was significant at least at the 0.05 level in all of those environments and combinations of environments. And the additive effect was surprisingly similar across all of those environments. And so if we add up everything, if we have this QTL in the homozygous state, we can get a 2 bushel per acre yield increase. So we hear a lot about yield QTLs are very location specific, very genetic background specific, and I'm sure in some cases that's indeed true, but I think this data shows that it's not true in all cases. What's interesting is the PI from which this QTL originated was introduced from China in 1926. So this pretty much predated plant breeding in both the US and in China. And I didn't realize until I was preparing for this presentation that I actually made the cross with this PI in 1979, the first summer that I worked for ARS. That's interesting. It's also, I guess, a bit frustrating when you think that it takes 35 years to accomplish some things. But when I started, I didn't have very much data, and I suspect that at that point, the year before I started, Clark Jennings was a soybean breeder with Pioneer, and he had started doing some evaluations of these PIs because they were interested in using them in their breeding program. And this was one that after a little bit of data looked pretty good, so I crossed with it the next year. 20 years later, we had just finished a major evaluation of a lot of relatively new soybean varieties from Asia, and then we wanted to go back and look at some other ones that we'd used in our breeding program, and so I put this one in and discovered then when we looked at this replicated data over years, it was 20 bushels an acre below our best checks and enlarged pretty severely. If I would have had this data in 1979, I never would have crossed with that line. And I think this raises some very interesting questions is about how do we select parents, especially from exotic germplasm. And not only was it low yielding, it was 26th out of 27 entries. It was at the bottom of the barrel. So the other thing we can do is to try to do genotypic comparisons. This last year, Perry Cregan and his ARS group at Beltsville, Maryland, completed the genotypic characterization of all of the annual accessions in our collection with 50,000 SNP chip. And then we ended up then with about 40,000 usable SNP markers on every accession. And this has proven to be a really valuable resource. A couple of things I want to say about that. We are one of few, if not the only, germplasm collection in the world that actually pure lines our accessions. Stephen mentioned most land races are heterogeneous. When we put things into our collection, we actually go through and we sort out that heterogeneity and we try to put them into pure lines, which makes it possible then for us to go in and invest a couple of $1 million to get these SNP markers because we know that all of these accessions are homogeneous and homozygous, relatively speaking, as nearly as we can get them that way. So this is a huge resource for us. And what we can do then is that we've gone in and then looked at the accessions out of there that we've used in our breeding program, and we've compared those then to conventional ancestors. So this is a clustering then where K is equal to 4. On the, over on, where's my, here we are. Over on this side, these are our 49 exotic ancestors that we've used in our breeding program that have all developed material that are, in most cases, are going to be equal or better than the best parent in that cross. And on the other side, the conventional are 47 conventional northern ancestors. And you can see there one big difference, that blue bar, are 4 wild soybeans that we have in there. There are some quantitative differences between the left and the right, which maybe indicate we have some different genetic diversity, but there's certainly nothing we can see here that makes it qualitatively different. If we look then at the population structure, and we did the same 50,000 SNP chip on our experimental lines, and on this side, oops, I don't want to do that. You can see these are our alternative lines as opposed to the conventional. And the conventional are all of the public varieties, well, all of the varieties in our germplasm collection, a few privates, released since 1995 in maturity groups 2, 3, and 4. So again, if you use your imagination, you might be able to see that perhaps we're preserving more genetic diversity in our experimental lines. And it might be a little easier to see when we break these groups down instead of by line as by groups. You can see with our alternative gene pool, more of our bands are of equal width. But we still don't have, I think, the kind of definitive answer that we're looking for in terms of have we integrated something that is really new. So the second part of the primary gene pool then is the wild soybean. This was some work that we worked with Perry Cregan, in which they sequenced several years ago 102 random genes. And then we compared them to a group of wild soybeans and then some Asian primitive varieties or land races. And this data would indicate that in the domestication process, we've lost at least half of the genetic diversity. So that would indicate that this may be a rich source of good genes that have left behind. If you're not familiar with the wild soybean, this is what it looks like. And I think you can see why there are a lot of genes in here we wanted to leave behind. This is when it's very happy. We have to grow these in cages because they're insect problems that don't at all affect our normal soybeans that cause great pain for the wild soybean. And you can see its seeds are about 1 to 2 grams per 100, whereas the normal soybean varieties are somewhere around 15. So they're very small. This is what the F1 looks like. So even after that cross, we haven't made a lot of progress. But these are lines that we developed from the wild soybean, but it took 1 or 2 back crosses to get to this point. And all of our back crosses were made to Williams 82. This is the line that was originally sequenced, and so is a good research tool. We developed BC2 lines from 4 different wild soybean accessions. And if you see here, most of these, there we are, under maturity. This is 3 days later, was significant. These are a day earlier, which really is not significant. All of the yields that I'm going to show you here, even though some of them are numerically higher than Williams, none of them were significantly different, either at the upper end or the lower end. And these are the donor parents. And over here, the percent is actually the percent of SNPs. In this case, we genotyped all of these with 1,500 SNPs. And you can see for these top BC2 lines, they were 6 to 9%, which is a little less than the 12% if they were unselected. But this bottom line, a BC2, we actually had 30% of the SNPs derived from the wild soybean. And then we had a set of BC1s. And what was absolutely remarkable to me was that, oh dear, If you, I think I'm poking this. One of those BC1s was developed by crossing two BC1s. And so we have the contribution of two different wild soybeans. And the total number was 56% of the SNPs were derived from those. The other BC1 had 24% of the SNPs. And if we looked overall, these are the number of lines that we had from each of these donors of the BC2s. And you can see that the percent of soja SNPs in there ranged from the low single digits to 20 to 30%. What was remarkable is that the average number of SNPs there between 11 and 19 was not significantly different from 12%, which you would expect from a regular backcross without selection. There was enormous selection for these. Selection pressure in each of these generations was probably 1% or less to get to the kind of viney plant that we saw initially to something that looks like a soybean. The other thing that we wanted to do is we thought, well, certainly if we went and looked to see where these SNPs were, we would find some major gaps where these traits for vininess and shattering and hard seed coat and all of those traits that separate the soybean from the wild soybean, those certainly would have been eliminated. And there was nowhere in there that we could find any major gaps where you'd expect these selection sweeps to have occurred as we selected very strongly against the wild soybean trait. So from that then, we became very interested in knowing where are these domestication traits and what's happening in these areas where you would think the selection sweep should occur. So working with Jianchen Ma at Purdue University, we developed a wild soybean by soybean mapping population. We've got 3,000 F6 lines. And we wanted to reduce that number for a replicated study. And so what we did is we measured about 7 or 8 traits that we could on here. And then to try to maintain maximum diversity, we would look at each of those traits in pairs. And so in this case, I'm showing you a really good example. We had lodging, which went from totally upright plants to totally flat plants. And then we had height. We had some between 15 and 20 inches. We had some 30 to 60 inches. And then with each of those 4 categories, we would try to pick lines. And you can see we were very successful here. We tried to get at least 20 lines in each of those categories. In some cases, we would get more because some of these plants would represent more than one category. So we're hoping then by doing this, we're going to maintain most of the variation that we had in the larger population and something that would be a little more manageable to evaluate. So this is what our population looks like. You can see, I can bring the marker up here without moving the, there we go. Some of these lines stand up pretty well. Some of them don't. You can see in the background those that are periodically standing up are our soybean parent. Now, one of the things when these crosses were made, we actually had a mix-up in our seed sources. And so instead of having one soja parent, we actually had 2. With our genotyping, we can separate those out. I think it may actually be an advantage and not a disadvantage in the end. But so you can see on the red and the blue arrows indicate the values of our 2 wild soybean parents, and the green arrow are our soybean parent. If we look at 100 seed weight, you can see in our population is very strongly skewed towards the small seeded, and we don't come anywhere near close to recovering the seed of the soybean parent. If we look at stem diameter, there is a slight skewedness to the wild soybean parent, but we can recover all classes, even some that are slightly larger, although not significantly more so than the soybean parent. And then if we look at plant height, the third category, we see transgressive segregation going in both ways. We see some very short plants and some very tall plants. So we've only started analyzing this data and just some preliminary results. This shows we get with a Manhattan plot doing our association analysis, we get a huge spike here for shattering. We would be really excited about this, except some Chinese scientists published this gene about 6 weeks ago. If we look at plant type, this is mostly the differences between vininess, and we can see this is probably going to be more of a quantitative trait, but we're also getting some very strong genetic signals. Now, Zhenshen Ma started working on some of the early inbreeding generations for these, looking at hard seed coat. And this spring, he narrowed that down to a region with only 2 genes, and then looking at the differences in those genes between the soybean and the wild soybean, actually identified the causal gene. And then we did in situ hybridization with the gene transcript, and you can see that with in the hard seed coat lines, this gene's is much more highly expressed in that very outer layer of the seed coat. So we've already now found one of the domestication genes, and we hope that we'll find more before the project is over. So there is no secondary gene pool for soybeans, but there is a tertiary gene pool, and this is made up of 26 perennial species. The wild soybean is native to East Asia. It occurs in Far Eastern Russia, in Korea, Japan, and most of Eastern China, although it doesn't go all the way to the south. The perennials are all native to Australia. So the theory is that perhaps there was some extinct common ancestor that has occurred somewhere, and then these got separated in geological time. The people that do such things estimated that the annuals and the perennial glycine were probably separated about 5 million years ago. So these could be a very good source of genetic diversity. The species that we're working with right now is Glycine tomentella. You can see that it doesn't look all that much different than the wild soybean. The flowers look like soybean flowers, but they're borne on these very long racemes. The seeds in the pods are very different. These are probably only about half the size of the typical wild soybean, and we can get maybe 5, 6, maybe up to 7 to 8 seeds per pod, which we don't ever see in soybeans. And the big difference, too, is that it has 78 chromosomes. So we're just short 1 pair of a tetraploid. So our wide hybridization, we can make the sexual cross between these two, but as soon as a cross is made, then things get very different. With the special hormonal spray, we can keep that pod on the plant for about 3 weeks, and then we have to excise these tiny seeds, and we put them on a medium. And what we want to do then is to introduce callus. It takes a lot of work to get these initial seeds, so if we do callus and then transfer it to a medium to introduce shoots, we can get multiple plants from a single seed. They develop, we then change the medium again, and we can get roots on these plants, and then we have our F1. But it's sterile. It has only 1 copy of the chromosome of each of the complements of chromosomes from the 2 parents. We treat that with colchicine. We get an amphidiploid then with 118 chromosomes. It's somewhat self-fertile. It produces a very few seeds, and it has the full complement of all the chromosomes, almost essentially a new species. It's not very good for much, except we can use it as a parent. So we back cross again. The BC1 now we recover all of the chromosomes from soybean, and we come with 1 copy of each from tomentella. And then what's critically important is the BC2, because now we have all of the soybean chromosomes, so they split normally. We only have 1 copy of the tomentella chromosome, and so when the mother cell divides, they get randomly split between the 2. And generally what we find is that we're getting between 15 and 19 tomentella chromosomes, so about half or a little less than half. And probably each one of these BC2 plants then has a different complement of chromosomes, and so this next back cross is almost the equivalent of making a new cross. I was curious, and so when I was preparing this, I went to find out that if you have 39 things and you take them 19 at a time, there's almost 70 billion combinations. So I think when I say that we have 12 to 15 BC2 plants and they're all different, the odds are in my favor. So then we continue back crossing until we get down to something that's 2n equal 40. This is genetically stable, it's self-fertile, and we can test it. So we just finished some yield testing, and I suppose as an introduction to that, when we first started this, we had no idea that we'd ever do yield testing. I mean, our idea was initially we were going to bring in some disease resistance because our Tomentella parent had that. To bring in something like yield from these lines, I thought was just way too ambitious to even think about. But we started testing. Some of them looked good. This is data from 12 environments over 2013 and 14. Dwight is our soybean parent. It's group 2 line. It was released from the University of Illinois about 17 years ago, so it's a pretty old variety, but it's one that works very well for what we are doing. But in this yield test, we have this line that, just the second one on there is essentially the same maturity as Dwight. It's 3.6 bushels an acre higher yielding, which is significant at the 5% level, and not different from Iowa 2101, which is our best check in the test. And it comes from this BC2 parent 06H1-3. Just one more example. This was a group 4 test, and actually our highest yielding lines were not group 4s. They turned out to be much earlier. You can see this line is a BC3. It's 6 days later than Dwight, but it's 7 bushels an acre higher yielding than Dwight, and from a different BC2 parent. So even within, if we were crossing elite by elite, to get yield increases of this magnitude would be surprising. To cross an old variety with something as diverse as a very distant relative in the tertiary gene pool is quite astounding. We tested 180 lines, and we found that 36 of them were significantly higher yielding than our recurrent parent. Now, most of these are also significantly later, so that's going to have certainly some impact on yield. We were then trying to determine, where were the genes that we had moved in, and so we did a genotyping by sequencing because there were no SNPs identified in our Tomentella parent. And usually with that procedure, we can get tens of thousands of SNPs. The problem here is that for Tomentella, we don't have a reference genome. So the only way we could call SNPs is to find those segments of Tomentella that would match up reasonably well with our soybean reference. And that limited our numbers very dramatically because the Tomentella genome is very different than the soybean genome. And then we also, because we were concerned about sequencing errors, we didn't use any SNPs unless it occurred at least in 2 different lines. So using those filters, we ended up with only 1,200 SNPs. But surprisingly, they were pretty evenly distributed over the genome. So given the fact that they are very different, we still got markers on all of the chromosomes and fairly evenly divided. We did our association analysis. We found significant effects with yield on 6 chromosomes. This was very exciting until we discovered that every one of these associations was with low yield. I'm not talking just a little bit here. I mean, most of these were 3 to 4, as much as 5 bushels an acre reductions in yield. Now, when you think about it, out of these 180 lines, we had most of them were equal to our recurrent parent. we had twice as many that were higher yielding than lower yielding, and yet all of our associations were with low yield. So originally, we weren't quite sure what to make of this data, and we're still not quite sure what to make of this data. It could be that there are some epigenetic effects when we're integressing two very different genomes. What I've sort of want to believe now is that this is at least evidence that what we brought in is really different from Tomentella. Because if we would have introduced segments that are similar to what's in soybean, then they should have matched up to our reference genome and we should have found SNPs. If we're bringing in segments that are very different than the soybean genome, they wouldn't match up, we wouldn't find those SNPs. And so the fact that we have lines that are much higher yielding, the fact that we don't find any SNPs associated with those, at least if you want to be optimistic, would say that that's because those regions are coming in from Tomentella and they are genetically very different. We're still going to pursue this to see what we can find, what we've introgressed. So in summary then, our commercially used gene pool has served us very well. We've been making progress in it for 80 years, and we're still making progress. But we're not making progress at the rate that we should be. Our breeding progress is not equal to our resource input. So we need to do something different. Using exotic germplasm is not high risk anymore. Those of us that have been working with it for 3 decades have now brought this material to the point where it's at least as good, if not better, than the best varieties out there. So it seems like an excellent opportunity to bring in new material that's genetically different with very little risk. But it still seems to be a hard sell. I think, well, for 35 years, I have been crossing with exotic germplasm to increase yield. And for all of that time, I thought it was a good idea. It's only been within the last 5 years that I've been convinced that it's a good practice, because that's when we get the kind of data that I've shown you today. 10 to 15 years ago, we were releasing germplasm derived from exotic material when we got to within 5 to 10% of the best check, because we thought this was different and that was good. Now we are routinely getting things that are 5 to 10% better than the best check, and that best check has gotten better over those 10 to 15 years. So again, it's a little direct evidence, but I would present that to you as saying, if we're using exotic germplasm, we're making progress faster than people are using the conventional germplasm. And that's what we need to do. So genotypic explanations would be very helpful. We don't know, we can't point to something yet that says, this is why we're getting this yield increase, this is why it's different, and that's what we still need to do. Once we have that, then we could start selecting on that in our germplasm collection. If you figure there's about 100 lines out of our collection that have been used in the commercial gene pool, over the last 35 years, I've evaluated progeny from about 500 accessions. There's still probably 100 in my breeding program and about 50 that have made a really significant contribution. So that means that we still have somewhere around 15,000 to check. And doing those 535 years is going to take us a long time to get through that. But if we can start to select genotypically and not just phenotypically, we can do that faster and we can do that smarter. And I think then that's the future challenge. How do we select parents? How do we go into our germplasm collection and figure out which of those 15,000 that we haven't looked at might have that next yield QTL that we may or may not find by putting it in yield tests, but could make a significant contribution. So then I need to thank a lot of people that make this possible. My group at Urbana, Ed Johnson, is a support scientist who makes possible all of the yield test data that I show. And Ram Singh, a special acknowledgement of him. He is a research geneticist now with ARS. He is the one who developed the procedures for making the crosses with Tomentella and developing the fertile progeny. And as far as I know, is the only person in the world that can do this. And I think his work has unleashed some incredibly valuable germplasm. Abraham Akperty is a grad student that did the yield testing on both the perennials and the sojas, and Stephen Swarm is working on the mapping with the wild soybean. Perry Cregan, David Heighton, and Chi-Chen Song did the SNP work. And then my long-term collaborator at Illinois, Brian Deers, and his graduate students, Carol Fox and Keith Rinker. Carol worked us together on confirming that QTL, and Keith on the decade study. Matt Hudson, Yuanjin Feng is working on comparing the alternative and the conventional gene pool, and Mohammed Balavov has helped us on the sequencing of the Tomentella. And Pat Brown and his postdoc at the University of Illinois are our go-to people for the genotyping by sequencing. And then Jianshan Ma is working with us on developing the wild soybean by soybean population, and Yuanjun Sun, his postdoc, did the mapping work to identify the hard seed coat gene. And then Grover Shannon and Andrew Skabu at the University of Missouri, and George Graff at the University of Nebraska, and Rolf Mian, ARS at Worcester, have been a big help in testing the material in the field to get the data that we have today. So North Central Soybean Program and the USB and ARS have provided the funds to make this possible. And with that, I will take questions.
Thank you, Dr. Nelson. I have a question about your looking into the future, about the resources we have to evaluate those germplasm in the seed bank. You talked about, I think you calculated the number of years for you to finish all the collection. And then you talk about the possibilities of genotypically evaluate those germplasm. The question now comes back that every time we try to genotypically evaluate or generate a prediction for those germplasm, the issue is that the base of that prediction or evaluation or value assigning process is based on some phenotypes where we have. That phenotyping process also has its own limitations. What do you think about that, actually?
I think you're absolutely correct. One of the things, actually, we're working on a project with George Graff in Missouri now, and we're taking the 50,000 SNPs, and we've used that as a model then to select within the germplasm collection, and we're taking those that are selected based on the differences. We're taking a random sample, and then we're going to be over the next couple of years doing some fairly extensive field evaluations of that material. And to see whether we can find things with the combinations of the SNP data and the phenotypic data that we wouldn't have found with either one alone. But again, the example that I used with the QTL, I don't think there's any way we would find that if that one ends up being at the bottom of the yield test. So that's where, if we could ever find a yield gene, and if we knew what that was, then we could start looking for variation, at that genotypic level. I'm assuming that there are some yield genes out there. They're pretty elusive, but I think what's very interesting is that with 80 years of soybean breeding, we've quadrupled our soybean yields. But we haven't the slightest idea why that's happened. So, if we had some idea about that, and if we had some idea about what we're bringing in from our exotic material, I think that would be very helpful in trying to go back into the germplasm collection.
Dr. Nelson, I'm not sure whether it's realistic to initiate a soybean conversion program like in sorghum.
I'm sorry?
Soybean conversion program like in soybean to convert the exotic lines to something more cultivatable to the U.S. Thank you.
Should we do a soybean conversion program? I don't know. That seems like a lot of work to me. And I suppose, most of our germplasm is really accessible to us now. We have material groups 9 and 10, which we grow in Costa Rica, which, we really can't grow here, but that's a very small percentage. Most of the others, we could cross. I think we don't have the kinds of extremes that I think we saw about earlier this morning. So I think there are better ways that we could get in that diversity rather than back crossing a few hundred lines.
Looking at your Manhattan plot, you were sharing your thoughts about some disappointment that this Manhattan plot pointing to the loci where those exotic materials, offering this lower yielding allele. And then I have some thoughts in my mind that isn't that because when you're doing the backcross, that generations of backcrossing wasn't enough if we follow the taxley strategy or advanced backcross QTL, that later generation you might be possible to release some of the potential allele effect instead of the earlier generation, or if whatever you have already in terms of analysis, we know those loci that have major effect going to be detected in the first place. But if you control those Manhattan peaks first, and you rescan the genomes again, you might find something remnant, smaller effect QTLs that has alleles that have possible favorable alleles from the exotic. Those two questions are somewhat connected. Thank you.
Yeah, I'm not quite sure I followed that, but if I don't, come back to me on that.
The first one is that whether you, because taxley promoting this advanced backcross QTLs, that you have to wait until BC2 or BC3, and then generating the populations for mapping and for breeding combined. So I wasn't sure that it's just speculation, because the research takes money, time, but speculation is free, right? and then the second, we'll just ignore the second question. Just have first one. Yeah.
I think the first one, many of these lines that we're testing are already BC4s and BC5s. So, yeah, and so I'm not sure, that we see any, there aren't any obviously major detrimental effects that we're seeing because of lack of backcrossing. We've crossed these now onto, other material, and we're developing, more traditional mapping populations to see where we can, if we get a different segregation in there, can we track those intergressed regions? And then your second question?
So the second question is that somewhat, if you have major QTLs that dominate your phenotypic variations, you would not find those minor QTLs that are possibly contributing favorable alleles from exotics. This GWAS whole process is that it's adaptive. If you actually fix those, some of these peaks first in the model, rescan, you might have a chance to find something that is favorable alleles from the exotics.
Yeah,
speculation
good point.
Again.
And actually, I just got these data just a couple of days ago, and I know that we were doing, Abraham was doing that on some of these, and I don't know whether he put these in these QTL were put into the model, and then to see what else would happen. But that's a really good point. The other thing, which I was afraid somebody was going to ask me and I wouldn't know, but then I'll ask this question myself. What I'm curious about knowing now is to going back and seeing which lines those QTLs exist in, and see what they're actually, what their yield values are. And I haven't done that yet, but I would think, are we finding, are all of these, most of our, because these are, BC flowers, 5s and 6s, our SNP frequencies are very low. So it's also possible that all of the, these significant associations are really with those low-yielding lines. But yeah, Mark.
Yeah, Randy, in the beginning of your talk, as you looked at the history of yield increases in the north-central United States, you mentioned that although many more resources have been directed towards increasing yield in the last 20 years or so, that you suspect that because of the need for a lot of defensive breeding that things may have been held back. And then you've given us some really exciting stuff about the role of exotics in increasing yield. But looking forward, would you care to comment about the role of defensive breeding moving forward for soybeans? What role are the exotics going to be playing in that? Do you see some changes there?
Yeah, I mean, I think they'll always be important because we're, right now, I mean, we are, with commercial soybean production is very heavily dependent on a very limited number of sources for most of major disease resistance. So soybean cyst nematode is a big one. Probably over 90% of all of the soybeans deployed that have resistance have it from a single source. That source seems to be doing pretty well, but we can't expect that to last forever. So, and I think you can name your favorite disease, and the situation is going to be similar. One of the things, I didn't talk about this, but from this tomentella, we've transferred SCN resistance. We're trying to figure out how different it is. We've transferred rust resistance. We're trying to figure out if that's different. We transferred Phytophthora resistance. And the very interesting thing there is that the first gene we found was RPS1K, which is the most widely deployed gene in soybean resistance for Phytophthora. At least it was a few years ago. So here we've got these two species separated by 5 million years, and we bring in a gene that's at the same location. We don't know about its sequence, but certainly by function, it's identical to the one we got out of soybean.
Thank you, and let's thank all of our morning speakers one more time.